Today's WALS lecture is the special NIH Director's Lecture, and that Mona was nominated by Women's Scientific Advisors Committee. Mona, before joining faculty at Princeton University, Mona obtained her undergraduate at Harvard University and then did her PhD in MIT, which she followed with a postdoc at Whitehead Institute. So, as we all know, we live now in the era of big data. Everybody is fascinated about using machine learning, deep learning, and all the learnings. However, I have to say that Mona was one of the pioneers of using machine learning in computational biology. So she pioneered quite a few methods, especially starting with approaches to predict DNA binding specificity. She developed quite a few of methods for this purpose, including binding specificity for coil-coil. The other was cis2, his2 zinc fingers. And especially I like this work, since not only it gave a wonderful understanding of binding specificity, but then it was also used in several very important papers, including paper in Science, which used this understanding and modeling using Mona's method to understanding the evolution of recombination hotspots. Later, Mona also played pioneering role in developing computational method for networks and system biology. She has a seminal paper where she used a network biology for functional prediction of gene functions, and also a method, a very fast clustering method, and also network-based approaches to cancer. Mona has a number of very distinguished awards, including President's Early Career Award for Scientists and Engineers. She is a recipient, or she's an elected member of ISCB, International Society of Computational Biology. She's also an elected member of Association of Computing Machinery, and this is for biologists in the audience, this is really one of the most prestigious associations for computer scientists. She's been also a very active member of computational biology community. She was chairing many of key computational biology conferences, RECOMP, ISMB, and many others. She's been also helping being a chair of mathematical modeling NIH study section. Well, I'm sure there's much more what I'm missing now. But I would like to add to this that also she mentored really a lot of great new young scientists, which are now having their own independent career all around US and world, and also including here at NIH. Now, I've known Mona for many years, and in fact, I have to say that she was the one who attracted me to come and join computational biology field. And she was the first one to tell me that there is actually a difference between DNA and proteins. So I was ashamed to admit that. but it's really wonderful to have her as speaker, and was always wonderful to have her as a colleague. Mona.
Thank you, Teresa, for that very kind introduction. It's really my great pleasure to be here today and to tell you about some of the work that my group has been doing in deciphering cancer genomes and networks. So mainly, I'm going to tell you today about work that is by Shilpa Kobrin, Borislav Hristov, and Dario Gersi. So very broadly speaking, my group develops computational biology methods, or method development group, to try to decipher genomes, in particular by thinking about the networks that bring genomes to life. So on the one hand, we develop methods for predicting various aspects of interactions. So we've developed methods, as Teresa mentioned, for predicting zinc finger protein-DNA interactions, different kinds of protein-protein interactions, and protein small molecule interactions. And on the other hand, we've also developed a number of methods for trying to analyze large-scale biological networks in order to learn something about how functions are accomplished in the cell. In the last few years, we've really started to think about these genome interpretation problems in the context of genome variation. So, of course, we're now in an era where we have lots and lots of genomic sequence data, not just for human, but for many humans. And, of course, these numbers will just continue to grow. And so how do you kind of decipher these genomes and explain how the variation in genomes leads to this great phenotypic diversity that we see? And in particular, we're kind of interested in how this diversity may lead to disease. So, of course, we've thought about these problems of genome variation and trying to understand variation in genomes, again, from the context of networks. So it's really in this broader framework that we've started to think about interpreting cancer genomes. So I think we all know that cancer is a disease where our own cells acquire mutations that allow them to grow in an unproliferated fashion. And people realized a long time ago that maybe it would be insightful, oops, I went the wrong way, to sequence these cancer genomes and try to uncover which alterations are seen in these tumors, and that maybe this was something that would help us explain how cancer arose and also maybe play a role in therapeutics. So you can sequence the tumor, you can sequence a normal blood sample, and then you call the differences, and these are the somatic mutations that have occurred in cancer cells. So, of course, we're now in a place where this has been done at large scale through efforts such as the TCGA and ICGC, where we now have tens of thousands of patient samples that have been sequenced across tens of cancer types. It's also well known, though, is that if you look at any individual tumor and you look at the number of mutations that you see, here shown in mutations per 1 million base pairs, and each dot is an individual's tumor, and this is split up by cancer type, you tend to see a lot of variation in the number of mutations you see per cancer type, and you also notice that you'll see many, many mutations that can occur. So there are lots of mutations per cancer genome, and yet only a few of these mutations within an individual are relevant for his or her cancer. The situation is even more complicated because the mutations that you see across individuals can vary quite a bit. So cancer genomes are heterogeneous. And this is true at the whole genome level, but it's also true when you look just at the gene level. And so for this talk, I'll really be focusing on mutations that occur, somatic mutations that occur within protein coding regions. So in this case, you'll see here are some number of individuals, and you look at the mutations that hit genes, and again, you see this wide variety of genes that are being hit. So how do you look at all this cancer genomics data and try to understand from the sea of mutations that you observe, which are the ones that actually play a role in either cancer initiation or cancer progression? So which are the mutations that drive cancer? So when cancer genomics data first became prevalent, people realized it would be really useful to look at this data at the population level. So look at a number of individuals together and aggregate the mutations that are observed in these individuals' tumors by gene. And so then you're going to look for genes that accumulate many mutations. The trick, though, is what is a lot? And so a lot of time was spent thinking about models for statistical significance. And the idea is you're going to try to highlight those genes, so in this toy example, gene 4, that's mutated more frequently than expected by chance, according to some background model. So indeed, these have been very powerful methods, and you can see all they leverage is statistics. But I'd say, while these methods have been very powerful and very successful, if you look at genes and you rank them by their overall mutation frequency across a population of tumors, you're going to see a picture like this, where there are a few genes that are mutated at high frequency, but there are many genes that are mutated at lower frequency. And indeed, among the genes that are mutated at high frequency, you're going to have many well-known cancer players. But if you look even at genes that are at moderate or low levels of somatic mutation frequency, you'll see some known cancer driver genes there as well. So I would argue that these frequency-based methods, though powerful, are insufficient. So I want to talk today about methods that try to first discover these somewhat less mutated yet genuine cancer genes that are sitting in this sea of genes with low mutation rates. And two, I'd like to start to think about the mechanism of action of individual mutations, to not just treat this as a purely statistical problem. So I think it makes sense to not just look at cancer data by itself, as these frequency-based methods do, but instead look at it in the context of all this other types of data that has been collected over the years about proteins and genomes. So for example, we have a lot of information now about protein structures. Within individual protein sequences, we often know which are the pieces of them that are the working parts, the most important parts. We have lots of data now across genomes, and that may inform our predictions. We have lots of data on population, natural variation data. And we also have large-scale biological networks for human. And so when we think about interpreting this cancer data, we want to think about these other data sources as well and see if we can put this data together in a way that would inform our idea of which genes and which mutations are the ones that are causal for cancer. So we're going to look at a lot of different data sources, but as I said, my group is really interested in networks. And so one thing that's sort of kind of driving our work is this idea that if we're going to look at genes and the proteins that are encoded and the mutations that hit them, we want to think about them with respect to the interactions that the proteins make. So proteins, in order to accomplish their many functions, interact with many different types of molecules. This includes proteins interacting with DNA, with RNA, with ions, with small molecules, and with other proteins as well. So if we think about these interactions a bit more, then we can decide that this would be a kind of a great framework with which to interpret this cancer mutation data. So today I'm going to tell you about two types of projects we're doing. The first is thinking about these interactions and trying to identify mutational patterns that change interactions. And then as a complementary approach, we're going to talk about somatic mutations in the context of networks and see if we can use networks then as a way to interpret these mutations. Okay, so going back to protein interactions. So if you look at a protein sequence and you look at its 3D structure, not all of the amino acids within a protein participate in its interactions. So in this particular case, the residues that participate in this interaction with this ligand are shown here in red, and you can see they're proximal in 3D. If you look at the 1D amino acid protein sequence, though, you'll see that the amino acids that participate in these interactions are scattered throughout the protein sequence. So if we want to think about mutations then that hit this protein sequence and whether they affect functionality or affect interactions, we have a good idea of which of these amino acid positions are more important. So in particular, mutations that hit one of these red amino acids that participate or that mediate this interaction with the ligand are more likely to somehow affect the interaction of this protein, either by disrupting its interaction with the ligand or acquiring new interactions. Okay, so we're going to look now at trying to interpret cancer somatic mutation data by thinking about protein interaction sites and where on a protein sequence the mutations are landing. Okay, so our overall hypothesis is that cancer cells malfunction due to mutations that change interactions and thus cellular networks. And our overall goal will be to try to predict cancer genes by identifying those with an enriched number of mutations in interaction sites across tumor samples. Okay, so how do we do this? Well, there's a kind of technical problem here, is we're going to look at all the genes in human, and we want to look at each gene and think about where the mutations are falling with respect to interaction sites. But it turns out that as of today, or as of recently, it's only about 13% of these human genes have a co-crystal structure where you have a complex or a structure of this protein in complex with a ligand. So we'd like to bring this number up. So how can we infer protein interaction sites across more human genes? Well, the first step is kind of obvious, and that's to use homology modeling. So maybe we don't have a structure for the protein we're interested in complex with a ligand, but maybe we have a structure for another protein that's highly similar to it, and we know which residues within this highly similar protein are in contact with the ligand because we have a structure for it. So Dario Gersi in my group built this pipeline where he mapped mutations onto protein sequences, then he did homology searches to come up with structures where we knew which amino acids were in contact with either nucleic acids, small molecules, ions, and peptides. And he used very conservative thresholds, greater than 60% sequence identity and greater than 90% identity in the binding residues, to transfer information from this structure to the sequence that we're actually interested in. So if you do this, you can raise the coverage of protein sequences where you have some information about interaction sites from 13% all the way up to 27%. So this is a step in the right direction. But we wondered, can we do better than this? Can we infer protein interaction sites across more human genes? And it turns out we can if we think about the building blocks of proteins. So proteins are built up of domains. So domains are repeating subunits within a protein sequence or across a protein sequence. And they are the fundamental unit of protein structure, function, and evolution. So one nice thing about protein domains is that protein domains can be elucidated from sequence data alone. So here's a cis2, his2 zinc finger domain, something that we studied a lot in my group. It's a DNA binding domain, and it can be easily recognized by looking at protein sequences just by using this probabilistic model to find matches to this model. So protein domains are ubiquitous. About 90% of human genes contain domains. And so our overall idea is that we're going to model how domains interact. And based on that, we're going to infer interactions in proteins with domain matches. So what we're going to do is, again, start from crystal structures, co-complex crystal structures, and we're going to use this resource BioLip, which has 25,000 proteins, 83,000 protein co-complexes across a range of different organisms. And we're going to put this together with domain information. In particular, we're going to identify domain matches within these protein structures, and these are matches to PFAM-based domains. Okay, so how might this work? So let's say we want to build a model of interaction for the BZIP domain. The BZIP domain is another domain that binds DNA. We're going to find all BZIP domains in BioLip, and we're going to aggregate them. And so here are your various sequences with your BZIP domains. And you'll recall that the probabilistic pattern has a particular length, and we can think about each position within this BZIP domain. So now we're going to go and look at each position in the BZIP domain, and we're going to ask, how close is it to the ligand of interest? So in this particular case, the BZIP domain interacts with DNA. And so we can compute, as we go down the position in the BZIP domain, what is the distance in, for example, position 1 in this structure, position 1 in this structure, position 1 in this structure, to DNA. And what I'm showing here are box plots of those distances for both the distance to the DNA backbone and to the DNA base. And so when you look at a picture like this, where things are up close in this part of the domain and further away here, you can infer that this is the part of the domain that interacts with DNA. Okay, and so we did this not just for DNA, but for peptides, ions, small molecules, and various subsettings of this. So what we wanted to do, though, was to go from one of these box plots to just compute a single value from this distribution that tells us some sort of propensity of how likely a position within the domain is to interact with the ligand. And so we used just a simple measure here. We tried more complicated things as well. And so we compute this per domain position, per ligand continuous binding potential score, where this number is just the fraction of structures where the amino acid at a particular position is close to a ligand with some distance cut off. All right, so when we did this in a large scale. And so we determined the positions involved in interactions across more than 4,000 domains, and we call this resource the Interact Dome instead of the Interact Dome. Okay, and so then you can say, I have a domain, I know the positions by aggregating structural information that are the ones that are interacting with ligands. Now I can find domain matches within human protein sequences and transfer the information from the domains to the sequences themselves. And so that's what we did. So as I mentioned, up till this point, we had 27% of human genes where we had some structurally modeled interaction. I told you before that about 90% of human genes have a domain. And it turns out that about 2/3 of human genes have an interaction domain that we're able to model. So we were able to go from just sort of naively 13% of genes where we had some information about interaction sites to about 2/3 of genes. Okay, so now what are we going to do with this, going back to the cancer context? So what we want to try to do now is to uncover genes that have a significant number of mutations within their interaction sites. So we have here a schematic of a protein sequence. We have modeled interactions shown by these shapes. So the first two are modeled. And for each of those domains where we have modeling information, what we have is this 0 to 1 score that's a per position binding potential. And that these scores are shown here by these bars. So now we're going to do analysis again across the tumors in a cohort-based type of analysis. And we're going to see where mutations fall with respect to these positions. So let's say we looked at our data and we had 3 mutations and they fell in this second position of this binding domain. So we compute a score that's just the sum of the binding potential where the mutations land. So in this particular case, our score would be 3. So we say, is 3 a big number? Is 3 a small number? How do we know of the mutations that fall within this region that we've modeled? Do they tend to hit binding positions or not? So the standard thing that you would do is you would say, well, let me just shuffle these mutations, and I'm going to shuffle them across the domain, and I'm going to repeat this multiple times, and I'm going to use this to build this distribution, and I can compute a mean, and I can compute a variance. So you can do this empirically, but it turns out that you can also just do a little bit of math and do this analytically and compute this mean and variance. And then you can say, well, I can take my score and use this computed mean and variance to compute a z-score. So I'll just say the fact that we switched from this empirical shuffling, which is sort of what most people do, to this analytical way ends up giving you huge speed-ups. So you actually get a 7x speed-up per shuffle, and you typically do 1,000 or so shuffles to get good estimates of these means and variances. Okay, it turns out that for this particular step, this speed-up isn't as important as it'll be in what's coming up. Okay, so now let's go back to our sequence. So maybe we've modeled this region, and we're going to ask, when we look at the somatic mutations that fall within this domain, do they tend to fall positions that are more likely to participate in interactions? And so we compute that. And then we also maybe model this region, and we ask the same questions of the mutations that fall in this region or this region. Do we see an enrichment in positions that participate in interactions? And as I mentioned, we now have modeled interaction sites within 2/3 of human genes. But we realized that this machinery that we've developed doesn't have to be specific to interaction sites, that we could also think about trying to uncover significantly mutated functional positions. So there are other things besides interaction sites that are important for protein functionality. So for example, maybe we want to know, when we look at mutations that fall across this entire sequence, do we see an enrichment of mutations that fall in this particular domain, maybe a kinase domain, or maybe a DNA binding domain? And so we can compute enrichment for mutations within these important regions, for example, domains. And domains, as I mentioned, cover 90% of human genes. But then we realized, actually, we do have measures of full sequence importance, and that comes from conservation. So you can look at the human gene, get homologs across different organisms, and you can look and compute conservation values across all positions, because we all know that the positions that are important for protein function tend to be conserved under functional constraint. Okay, so now we have another type of track where we have a 0 to 1 score for every position across the protein sequence, and of course we can compute this for all of our genes. And we can ask, when we look at the somatic mutations that fall within this gene, do they tend to fall in conserved sites? Next we thought, well, maybe we can use the same framework actually to include whole genome rate. So I told you at the beginning, there's a very powerful signal that says if a gene is hit more often than you expect by chance, it's more likely to be a cancer driver gene. So the likelihood that a gene is mutated turns out to be affected by many factors. Other people have shown this. So for example, if a gene is long, it'll tend to accumulate more mutations. Or if a gene is expressed at a high level, it tends to have more mutations. But let's say that we looked at all of our genes and we already know somehow this background per gene likelihood of being mutated. So where might this measurement come from? People previously have used very complex models to try to compute this. But Pavel Prstiki in my group realized that you can actually use the amount of natural variation you see across populations to set this background distribution. So many of the things, for example, sequence length, are still relevant here. And the overall intuition is that a gene that can acquire lots of common variants and across healthy populations is also able to acquire lots of somatic mutations without affecting the functionality or breaking the functionality of the gene. So we used natural variation across the human genome to set these background per gene likelihood of being mutated. So now we can look at the mutations that fall on any of these genes and use this as our background distribution and just compute whether the number of mutations that hit a particular gene are more than you expect by chance. So kind of putting it all together, we look at natural variation data, which lets us take into account whole gene mutational frequency. We look at conservation data, we look at domain data, and we look at interaction data that comes either from homology modeling or from our domain-based method. So the next question is, how do we put together this very different sources of information? Each one is asking the same basic question, which is, are we seeing an enrichment of mutations in pieces of interest? So this turns out to be non-trivial because, as you can see, these different tracks overlap each other. So if you look at the mutations that fall within one track and another track that's overlapping it, the background expectation is that there's a correlation there. So what we figured out how to do, and we built this system called Pertinent for perturbed and interactions and also for pertinent cancer genes, where we built a background model where we analytically computed this covariance matrix between these multiple different sources of data. And so then we use this covariance matrix to set up a background model, which is the standardized multivariate Gaussian. Then we looked at the actual mutational data that we saw across tumors, and we saw whether they were enriched with respect to each of these different information sources. And then we see where that fits with respect now to the standardized multivariate Gaussian. And that's our overall scoring scheme. Multiple sources of data, which we're then able to put together. So now we can think about evaluation. How does such a method work? So one thing you can do is now you score all your genes, looking at all your data together. And we're going to look, as we go down our list, whether we see an enrichment of genes that fall in the cancer gene census, which comprises a list of genes that have been causally implicated in cancer. So we have multiple sources of data, and we can look at them independently. Okay, and so this is across 33 cancer types, 11,000 plus tumors that we're looking at. So if you just use the natural variation tracks, you can see that there's this clear enrichment for known cancer driver genes in the top of the list, and this goes down. And of course, it would go down all the way as you go further down the list. Okay, so that's your classic frequency-based method, with our twist being that we get our background values from natural variation. You could look at just the conservation track, where all you're asking is, if I look at the somatic mutations that hit a gene, do they tend to fall in conserved residues? And you can see that that, too, is predictive of whether a gene is a cancer driver. So again, you see an enrichment of cancer driver genes in the top of the list that's maintained as you go further down the list. You can do the same thing asking, do I tend to see an enrichment of mutations within functional regions such as domains? And again, you see this same pattern. And then you can look at interactions and say, what if I try to compute whether I have an enrichment of mutations within interaction sites? And you see this nice kind of especially precision at the top of your list. And then you can ask, what happens when I put these different data modalities together? And what you see is that you're able to benefit from these complementary sources of data. So as I mentioned, this was 33 cancer types, 10,000 tumor samples. And I wanted to say that this whole process takes 10 minutes to run on a standard, or at this point, weak desktop. And the only reason it's able to run is because we're able to do the covariance calculations analytically instead of trying to do it empirically and compute that for all pairs of tracks. All right. So one thing I said was we wanted to be able to use methods like this that weren't just based on frequency to kind of pull in this tail of the distribution of things that are more lowly mutated. So here's a plot where we ranked genes by their mutation rate, so from the most mutated to the least mutated, and that's just the gray line that's shown here. We just highlighted the top 35 ranked genes by Pertinent. And we can see that, of course, we identify many highly mutated genes, and that's sort of shown up here. And of course, we're skipping some of these highly mutated genes. These aren't things that are cancer relevant, but they are mutated in high frequencies. But we're also able to pull out genes as we go further down, and we're looking at fairly low missense mutation rates. So we're able to dig into this long tail. Okay, so I went through all this machinery trying to figure out what the interaction sites were. And so I want to show you that there's actually a benefit to doing this, which is you can go back now and look at the hits that you get by running this method. And you can go back and try to figure out what was the reason that this gene scored high. And so, for example, you can see that IDH1, so on this side are the known cancer genes. On this side are things that we predict but aren't yet known to be causally implicated in cancer. And you can say, what is the putative mechanism, or what is the effect of the mutations that you see in IDH1? And you can see that, according to us, this is disrupted small molecule binding, disrupted ion binding. Or you can see that there are many things that are involved in small molecule binding. And so the way we organize this is we have the first part where we look to see which specific types of functionalities might be perturbed. And then the second part is if we couldn't pinpoint precise residues or positions within domains that were perturbed, we would just overall ask whether certain types of domains, for example, DNA binding domains or protein binding domains, had an enrichment of mutations within them. So by adding this infrastructure onto our prediction framework, we can go back and make some inferences about what we think are the effects of these mutations. Okay, so what I've told you about in the first half of my talk is Pertinent, which is this integrative framework for identifying cancer-relevant genes that uses structures, domains, cross-genomic information, as well as population information. And what I showed you, I hope, is that these perturbed interactions are predictive of cancer genes and can be used in these larger integrative frameworks. As part of the development of this project, we also developed this resource, the Interactome, which may have applicability for interpreting mutations that you see in many other disease types as well, because it allows you to see where mutations are hitting interaction sites. And as I mentioned, we get coverage from between 63 and 69% of human genes. Okay, so now I want to switch to the second type of thing that we've been thinking about. So the first type was really focusing on mutations and whether they disrupt or add new interactions. Now I want to think about mutations in the context of these large-scale biological networks. So what I've told you about thus far has looked at each gene one at a time. But it's well known that cancer is a disease of pathways. And so here's an example from the TCGA glioblastoma paper. And it's showing this signaling pathway that's altered in 88% of the tumors that were sequenced. But if you look at the individual components of this pathway, you see that they can be mutated in low frequency or moderate frequency or high frequency. And so this argues that we shouldn't just look at, for example, one gene at a time when we do this type of analysis, that maybe we should be looking at this at a larger scale of groups of related genes. So, as I just argued, proteins that are relevant for disease, such as cancer, often have related functions. If you look at protein interaction networks, which have been determined at this point for large, for human and other organisms as well, it's been observed over and over again that proteins with similar biological functions or that participate in the same pathway or that take part in the same protein complex tend to be proximal within networks. So here is the part of the network in the schematic responsible. These proteins play a role in response to DNA damage, and these proteins here play a role in cellular growth. And so the function, for example, cellular growth, can be altered by mutations in any one of the proteins that participate in this function, whether it's this one or it's this one. Okay, so the idea then is we're not going to look at the mutations that hit an individual protein one at a time, but instead we're going to look at mutations together with these large-scale biological networks with the idea that proteins taking part in the same functional process or pathways are proximal in networks. And we're going to add data, and so in this case it's an individual that has mutations in, say, proteins 5 and 11 onto this network data. And the goal or the idea is to identify neighboring proteins within these networks that together are mutated across a large number of individuals. All right, so this is trying to interpret now mutations in the context of these networks. So just as a schematic of what is going on, say you have these 6 individuals, and the proteins that are found to be mutated within them, somatically mutated within them, are given here. And they're in this little toy example network over here. So the first thing we would do is label each of these proteins, for example, TP53, by the individuals where that gene or protein is found to be somatically altered. So in this case, individuals 5 and 6. Okay, what we want to do then is try to identify a small subnetwork that covers most of these patients, meaning that most of these patients have a mutation in one of the genes or proteins that we output by our procedure. Okay, so there's a natural trade-off here between the size of the subnetwork that is output and the number of patients that are covered or whose cancers can be have a mutation in one of the genes that comprise these subnetworks. So you can see that if you choose all your proteins in the network, then you have high coverage of patients, but you're not really uncovering disease-relevant proteins. You're just splitting out the whole network. On the other hand, if you output a subnetwork that covers very few proteins in the network, you're going to have lower coverage of the patients. There are going to be many patients that don't have a mutation in one of the genes comprising this subnetwork. But this connected component is more likely to consist of functionally related proteins. So we developed this approach called NCOP, Network-Based Coverage of Patients, where our goal is to find a connected subnetwork, G' of the overall network, that minimizes something simple. So one part, this first part, is just the fraction of uncovered individuals. And the second part is just the size of the subnetwork that you're outputting. So you want to, ideally, you want to minimize the number of uncovered individuals while also minimizing the size of this subnetwork, trying to capture this trade-off, which we deal with a single parameter alpha. So you can write this more mathematically, and you can solve this exactly with integer linear programming, but it's time-consuming. So instead, you can do a very simple heuristic procedure that works reasonably well. And so your procedure is to try to build this subnetwork by picking a protein that is found to be mutated in a lot of individuals, and you'll do this probabilistically. And then you're going to expand this subnetwork out. So if you call this subnetwork you're building G', for each protein in G', you're going to examine its interactions and the interactions of its interactions. And you're going to add to this subnetwork the 1 or 2 proteins that improves your objective function, that had that alpha trade-off the most. And then you're going to repeat until no more proteins that are being added improve your objective function. And so as I mentioned, this has reasonable performance as compared to the exact optimization, but it's much faster. So we have this one parameter alpha, and it turns out that you can automatically set this parameter. So you could look at a crystal ball, but that's not really the best way to do it. So instead, we empirically assign this parameter. So we're just going to vary this parameter alpha between 0 and 1. And then we're going to withhold some fraction of the individuals, say 20% of them. And we're going to use our greedy procedure and find a connected subnetwork in this graph using 80% of the patients that you left in. So those are the patients that you're trying to cover. And if you've done a good job in finding your subnetwork, then it should also cover these 20% of patients as well. Okay. And then you repeat this process many times, and you're going to use that to select alpha. So what happens is you vary alpha from 0 to 1, and you look to see what fraction of the time the individuals that you're looking at have a mutation in the subnetwork that's being output by the greedy procedure in both your training set, that's shown here in blue, that's 80% of your data, or in your validation set, the 20% that you left out. And where these two curves start to diverge, you're spending too much time trying to explain the cancers of all the individuals in your training set, and you're overfitting to your data. So you can see at some point you keep putting more and more nodes into your subnetwork, but the performance between these two things, doesn't, the validation performance does not increase. Okay, so the overall framework is you look at somatic mutation data across individuals' tumors, you look at protein interaction networks, you put the somatic information data on top of this network, you use this procedure I just outlined to automatically pick an alpha, and then you use that alpha on the graph as a whole. So this is a randomized process overall, but we add a bit more randomness. And in particular, we select 85% of the patients. We find the connected subnetwork using these 85%. We just iterate this process 1,000 times, and in the end, we output the genes and we rank them by the number of times they're found in the subnetwork that's been output. Okay, and then we can do the same type of analysis that we did before, where we can look at a particular cancer type. So in this case, kidney renal cell carcinoma, 416 patients. We look at a protein interaction network. Here we use HPRD. We've tried other networks as well. And we use the score that's output by NCOP, which had to do with the number of subnetworks the protein or gene was found in. So we want to compare our method to other methods. Our method, again, was NCOP. We want to compare it to a version of itself that uses no network information at all. All it asks is, do I cover the patients? Okay, so we call that set cover. And we want to compare it to just a frequency-based method, so ignoring anything about the network, but just looking at the frequency of mutation. Okay, and so what we see is that our approach, NCOP, outperforms, as judged by the fraction of proteins in the known cancer set, the other two methods quite easily. And so quite notably, the difference between this red curve and this blue curve is the advantage that you get by including network information in this formulation. All right, so this is nice to look at one cancer at a time, but you can also do this, you can automate this, and you can look at the area underneath these curves to get a rough idea of how much better one curve is than the other curve. And so that's what we did next. We looked across 24 cancer types individually, 6,000 samples, and we said how much better is the area under the curve for the NCOP method as compared to the set cover method that ignores all network information, or the NCOP method versus a popular frequency-based method. And we can see that NCOP outperforms the set cover-based method on all 24 cancer types, and it outperforms the frequency-based method on 22 of the 24 cancer types. Okay, we can also, if we're not the first person or first group that's thought about putting together somatic mutation data with network data, so we compared also to a previous network-based method, and you can see that here we outperform this other method for 20 of the 24 cancer types. And so again, what's being shown is the change in the area under those curves, and it's the log-fold change. So it's not just, this shows that network information is important, but it's not just network information. This shows That is how we use the network information that's important as well. Okay, like our previous approach, we again can look at genes and rank them by their overall missense mutation rate. And we can see which genes are the ones that are output by our methods, and those are the ones that are shown here in red and blue. So red corresponds to known cancer genes, blue corresponds to our novel predictions, and we can see that we output obviously some highly mutated genes, as well as you can see things that are mutated at lower levels, and these would be genes that couldn't be detected by frequency-based methods. So we can look at, for example, the genes that are just in sort of the lower mutation rates. And what we would observe is that we see this subnetwork that together is mutated in 14% of these kidney cancer patients. It contains 8 known cancer genes, and it's involved as a whole in AKT signaling. The novel predictions that are included here, shown here in blue, are interesting. So for example, we're looking at a kidney cancer data set, and one of the proteins plays a role in kidney development. Okay, so what I've just told you about is this network-based coverage of patients to try to uncover cancer driver genes. And I want to argue that networks provide a context within which to interpret these per individual mutational profiles. And the overall idea is we're trying to find small subnetworks where many patients have mutations in at least one component protein, even if none of the proteins individually are frequently mutated across all the individual's tumors. And I showed that it's effective in practice. So in the last few minutes of my talk, I want to tell you a little bit about what we've been thinking about recently. And it's still thinking about mutations in the context of networks. So this second part of my talk, what I was trying to tell you about was looking at new experimental information, and it really was proteins that were found to be mutated in cancer patient tumors, and thinking about it in the context of networks. So there's another paradigm that people have used for looking for disease genes in cancer and other diseases as well, and that's been to look at prior knowledge and kind of look at these two things separately. So what we'd like to argue is that all this new knowledge that's being acquired, again, shouldn't be considered in isolation. We should also use this prior knowledge about proteins that are known to be cancer relevant to somehow guide our network-based processes. Okay, so this has been done in the past, looking at just prior knowledge by itself. So maybe we know that these 2 genes are relevant for some disease, and we want to try to uncover other genes that might be relevant for the disease. A very common thing is to kind of spread the signal from these known set of genes. And the type of information, the type of method I just told you about doesn't do that with known disease genes, but does that with new information. And again, kind of thinking that if we grow out from this new information of highly mutated genes, maybe we're going to find other disease-relevant genes. So I told you about methods based on coverage, but there are other methods that are really popular as well, and they're based more generally on techniques for propagating information through networks. And there are several well-established propagation techniques. I'll just tell you about one of them, really, and that's based on flow or diffusion. And the overall idea is that each disease protein is some source of flow, and you're going to be spreading this flow across your network. And so in this particular model, the flow diffuses uniformly over the edges or the interactions. And then each protein, as the flow enters, drains flow at a constant rate. And so the idea is when you do this process, things that are close to your initial set will tend to kind of have high scores, and things that are further away will have low scores. An alternate type of established propagation techniques is based on this idea of random walks, and we've looked a lot at random walks with restarts, but I won't tell you too much more about that. I'll just keep this pretty high level. Okay, so what we wanted to do, again, in line with the first part of our talk, is to combine this new information with prior information as well. So let's going back to the context of cancer. So if you have a set of known cancer genes, we know that these cancer that there are some well-known cancer pathways, and if you look at protein interaction networks, that the genes that comprise these pathways are proximal within these networks. So if you propagate information from these known cancer genes, you're going to build out from these genes to other genes that perhaps are also in related and relevant pathways. Okay, so you can propagate this information using these flow-based techniques, and you would end up with this network where the proteins are essentially colored darker if they're closer to your initial set of gold standard known cancer genes, and then they're lighter for proteins or genes that are further away from this initial set. Okay, so this propagation ranks proteins based on their proximity to the prior knowledge set, and people have shown in previous literature that this is predictive of disease proteins. Okay, now we also have new information. So for example, this could be the frequency with which this protein was mutated across cohorts of cancer patients. So we can spread this information as well. But we want to do this in such a way where we interpret this new somatic mutation data using what we already know about known cancer genes. And so what we can do is use this information to guide how new information is spread. So now, instead of just spreading information uniformly from this new information, we're going to tend to go towards these darker colored genes that are closer to the set of genes that comprise our prior knowledge. Okay, so we're going to propagate new information biased by this prior information. And so maybe we get a picture like this, where the darker greens now are things that are closer both to your new information and they're more proximal to your prior information than not considering this. Okay, so that's kind of our overall flow for this reasoning, is to try to look at not just new somatic mutational data on networks, but to combine it with known disease sets already. Okay, so we can think a bit about how to test this. So just as an example, so our approach is called UKIN, using knowledge in networks. I'm going to show you some testing on glioblastoma, 278 individuals. We have our set of known cancer genes, the CGC. We're going to fix a subset of this to be a test set. And then from the remaining genes, we're going to sample some subset of them to act as our prior knowledge. So this is a little bit tricky from the point of view of testing performance. And so basically, we want to test what happens as you choose different amounts of prior knowledge, different sets of proteins, different numbers of proteins, and that's why you want to fix this set that you're going to try to recover. And then you can repeat this many times, and you average. And so what do we want to compare? We want to compare our approach, UKIN, which has this prior guided propagation from new information. We want to compare it to an approach that just does unguided propagation from prior data. This is something that people have done for many years. And we also want to compare it to unguided propagation from new data, which is something that people have started to do in the last few years as this type of data has become more common. And then finally, just as a benchmark or just as a sanity check, we can compare against just ranking all genes by mutational frequency. And again, we're going to consider this type of plot where we consider a larger number of proteins, and we're going to ask what fraction of proteins are in a known cancer set. Okay, and so this is what this looks like. So blue is our approach, and that's shown here. Green is the frequency-based approach, which you can see does quite well at the beginning, but tapers off rather quickly. Purple is when you propagate information just from the new data. So you can see just how much of a benefit there is incorporating this prior data in your network propagation. And then finally, this is what happens when you just do unguided propagation from your prior data. So this is the case for glioblastoma, but again, you can look at the areas under these curves and compare them, take a ratio, take the log. And again, if you look across 24 different cancer types, you can see that this approach, UKIN, does better, so positive log two-fold changes in AUPRC, than on all the cancer types as compared to when you're doing an unguided propagation from prior information, unguided propagation from new information, or if you just rank by mutational frequency. All right, so I want to argue that our new approach, UKIN, shows that there's clear benefit to interpreting new potential disease genes in the context of previously known disease genes. So these, we should learn from what we know already. I mean, there's a big move in machine learning to do everything in an unbiased fashion, but we shouldn't ignore what we already know. So what I described in this last part of my talk was a general framework. I showed its application to cancer, but it's also applicable to other heterogeneous diseases, and we've tested a little bit of this. So your set of prior data comes from some gold standard, but your new information, for example, could come from GWAS studies, again, where you're going to have an enrichment of things that have low p-values that are going to be disease relevant, but not all of them are. And I wanted to add that this model can be extended in various ways. For example, you can have varying degrees of confidence in your prior knowledge. So I'm going to stop here. And so I told you about two types of ways in which we think about cancer somatic mutational data. On the one hand, we developed these integrative methods to identify perturbed interaction interfaces, and this was largely the work of Shilpa Kobrin and Dario Gersi. And on the other hand, I talked about network-based methods for discovery of cancer genes, and this was largely the work of Borislav Hristov. I also had contributions to the work I talked about today from Pavel Prosticki and my colleague Bernard Chazelle. So you can download these papers, and you can also download or use the tools that we've developed for this. And I'm happy to take any questions.
And maybe as people go to the microphone, I was wondering, did you think about combining your two approaches and in your notes that have prior information, put your information from...
Pertinent?
Yes?
Absolutely. I mean, that's our clear next step for what we're going to do. I mean, these projects have been developed in parallel. Mm-hmm. It's a bit of a moving target, but we're absolutely really excited about the Pertinent approach and how it scores genes. And so that would be used to guide some of the stuff that we're doing next.
I was speaking of tumors, but those of us with experience in pathology know there's essentially two big groups. One is pediatric pathology, where you get things such as neuroblastoma, Ewing's, Wilm, medulloblastoma, and Burkitt's, rhabdomyosarcoma, retinoblastoma, etc. And in adults, you get a totally different group of tumors. Mm-hmm. Now, what I'm getting at is, wouldn't you want to stratify this further by age? It wouldn't make any sense not to. If you look at the chart in terms of the number of mutations and age,
yes.
It perfectly corresponds
right.
to
So
pediatric.
So we haven't done stratification by age. We have done stratification by tumor type. So in the first part, I showed results for pan-cancer analysis, but we've gone through every, the 33 different cancer types and done the analysis individually on each one. I think it would be interesting to stratify it in different ways, stratify also based on subtypes even within that types of cancers or other ways. I think the limitation is with the amount of data that you have as you do the stratification. So essentially, even though we're adding all this biology in the end, we're looking at statistical signals. So I think that'd be a really interesting thing to try, and we should try it. And I think the cases where you have lots of data, so probably the older people, you're going to have more predictive power in what you find.
Okay. The biggest problem, or I don't know if it's how big it is anymore, but it used to be the cancer of unknown origin. It used to pop up now and then. Now, in terms of diagnosis, that's like the mother of all diagnostic problems. And there's been a lot of genetic kits that have emerged to try to solve this problem. For example, you have a lymph node here. Nobody can figure out what it is. Have you tried to apply this towards that or commercialize it in that direction?
I have not looked at that at all. I mean, so you're really talking about cancer signatures now, I think.
Yeah, it's a big thing.
I mean, I think it's very cool. We have not tried to look at that.
Okay, thank you.
So I can follow if you don't. So first about the heuristic approach. As you may remember, we had a follow-up paper that we tried to have a data mining approach to not solve the ILP, but still get the guarantee optimality without putting any bound on the running time. When you compared the approach, were you able to see that every time for every single gene you got the same every starting clue?
No, we didn't. I mean, we definitely didn't. So it's something that I think the fact that we repeat the algorithm multiple times actually adds a lot of robustness to it. So in the end, we sample out the patients that you see and run the process many times and then rank genes in the end by how frequently they're output. But no, you don't always, the heuristic's not that good, right? Yeah.
So I have a question. I've been playing around some with some of these cancer gene repositories, and I was wondering, do you have any sense of what might change in terms of important genes that are drivers if you look not just at missense mutations but also at nonsense mutations?
I should say we actually did look at nonsense mutations as well. So it's not relevant for the interaction site part of what we did, but when we looked at the whole gene mutation rates, we looked at nonsense mutations as well. And so it's well established by others that you tend to see, tumor suppressors that accumulate these nonsense mutations. And so there definitely is a different pattern of interactions that there definitely are different types of cancer genes that you would recover, but we actually looked at both. Yeah.
So I think we are getting signal to leave the room. So let's thank Mona again. And I think we have right outside.